Okay, I think we're ready to get going. Welcome back. 3-day weekends. Gotta love them. All right. Why can't they all be like that, right? So we left off briefly getting into chapter 9. We got a handful of slides into it. We're thinking about different ways that we can cultivate bacteria and ultimately try to enumerate, try to figure out how much stuff do we really have going on. There are lots of different ways that we can potentially try to figure out what do we have going on. We talked a little bit, I'm going to back it up quickly. We talked a little bit about some growth curves, trying to figure out rates of these whatever microbes, how quickly are they able to start reproducing. We looked at a little of that, and I talked about some potentials like using some spectrophotometers. We're going to see that here in a second, to make measurements, basically looking at how cloudy, how turbid that broth is, and that allows us to make some estimations. There's other techniques like these Petrov-Hauser chambers or hemocytometers, same but different, that allow us to make some counts with, again, broth culture that's applied. Staining to see viable versus dead cells, and then some culture counters that allow us to count, and this would also be in a broth suspension. So this is where we left off. And the idea with this is to do a series of transfers from the stock culture, our jar over here, our bottle, to do some dilutions with this and then plate them out, each of these dilutions, to see how many organisms are ultimately in this jar. All right. So the serial dilution, you take, and there are numerous ways you can do serial dilutions. All right. It truly depends on what you may be working with. In this case, all of our test tubes right here have the same volume. All right. And this would be whatever this crap's growing in, nutrient broth or whatever, likely the same liquid is in here. All right. And we take a known amount, we take a milliliter out of the jar. I like to call this the mother jug. This is the one that's got it all, right? So I take a mill out of this and I put it into 9 mils of broth, all right? And typically you're going to do a little bit of mixing to get it all kind of dispersed evenly. Sometimes it's pipetting up and down with the same pipetter that you sucked it up out of. You put it in there, pipette up and down, switching tips as you make your transfers. You can use a vortexer in the lab where you just push the tube right onto it and it shakes it real quick. You also, without that, you can hold the tube, not by the cap, but by the glass, and you can flick it with your finger a little bit. We call that finger vortex. And you give it a couple of quick flicks, and that can mix it up. Then you take a bit of that out, and we do that repeatedly. All right. So it's the same amounts as we're moving through here. It was a mill out of this into that, a mill out of that after it's been mixed, and we move through. So 1 mil into 9 gives us 10 mils total. This is a 1 to 10 dilution. 1 part of this, 10 parts total is what we're left with. All right. So we've diluted it, but barely. All right. It's still got a boatload of organisms, assuming this has a boatload of organisms, which it obviously does. All right. So each of these, after we've done the mixings, oh my gosh, are you all right back there? But they got you. Thank you. All right. So after was like, holy, there's a tragedy going on back there. So after we have done this first mixing, we take a mill of that out. We do all of these transfers first before we start putting them on the plates. All right. So this mill out of the 1 to 10 into another 9 mil solution makes it 1 to 100. We're just multiplying by 10. We call this a 10-fold dilution, every single one of them. All right. And I said a minute ago, sometimes we may do this different. It depends on how we really want to do these dilutions. Maybe you don't have a huge amount of stuff. Maybe we might start off with a 10-fold dilution, and then we might do 2-fold dilutions where we're just doubling it up. So 1 to 10, the next one would be 1 to 20, and then 40 and 80. If that maybe wasn't super turbid, maybe we wouldn't want to dilute it out so far. All right. And maybe we're not doing this type of stuff, but something completely different in the lab where we're doing some dilutions. There's lots of different ways that you can dilute out whatever it is you're working with. All right. That reminds me of old school stuff. I'm old, right? The solution to dilution, or the solution to pollution, is dilution. That's what they used to say. When in doubt, dump it out, right? All of that is stupid. You don't want to do that, right? Just flush it down the toilet, right? That's not what you should do. All right. I mean, you should flush some stuff down, but you shouldn't take your pharmaceuticals and dump them down the drain. All right. That's not the solution. The fish will thank you for not doing that. All right. Dilutions. Back to that. So 1 to 10, 1 to 100, 1,000, 10,000, and 100,000. By the time we get to this last tube at a 1 to 100,000 dilution, we have seriously diluted this culture. And the idea now is to try to plate it. So we don't take a full mill out of these tubes. We take 100 microliters, a tenth of a mill. All right. 100 microliters put onto this. And it truly depends on what we're working with. All right. Sometimes we will do these dilutions and you will have a tube that has liquefied agar, still warm, but not cooking hot. And you make your dilutions in this. After you've done that, you can put it on the plate, spreading it around, and it will solidify and grow. All right. Or you pipette the broth, no agar in this stuff. You pipette the broth and then you spread it around. All right. Pour plating and spread plating give you some similar stuff. It just depends on how you're doing it. What are you working with in that tube? Does it have some solidifying agent or is it just liquid? That's going to dictate what you're actually doing with it. All right. And there's different stuff that we may like. And I'm going to show you both of those. All right. So either way could technically be happening. Just depends on what's up in these tubes. All right. So they get put onto the plate and spread around, however. All right. And then we incubate them, pop them into the incubator for a day or two, and then pull them out and look at them. And truly what we're trying to see are some individual colonies. We'd like to be able to count this stuff, all right? There are likely going to be numerous plates that you're not going to be able to count. It's just like a sea of bacteria, potentially not even seeing any individual colonies, what we might describe of as a lawn of growth, all right? Just completely covered. That's this guy, all right? Too numerous to count. Those are good ones, right? You're like, sweet, don't have to deal with it. TNC, right? Too numerous to count. Same with this one. I can see some individual bits in here, but I would not try to count this. This next one, now we're getting to something that technically is countable, all right? I don't want to count this, but I might want you to count it so I don't have to, right? Wouldn't that be the way to do it? There are devices that allow you to maybe do this a little bit better. You can put it on like a light box. Sometimes the light box has some gridding under it, kind of like that hemocytometer I showed you a second ago. And then you can start making some counts. There are also some nice little Sharpie pens that are electronic. And every time you touch a colony, it leaves a Sharpie mark. So you know you already hit that guy, but it also counts it on your pen. And then you know, so you don't have to keep track in your mind. You're just touching dots, right? And then you look at, and you're like, oh, sweet, 389, right? That's good, right? I imagine situations when you're halfway through counting and then someone comes in and starts talking to you and you're like, damn it, I'm counting, right? And you start over, right? So those little pens may help with that. So this is a boatload of colonies still, 389. You might be able to count it, all right? But there looks to me like a lot of stuff growing super close together, and I'd be hard pressed to say, are there 5 or are there 15, right? This next one, though, with 50 colonies, that sucker's countable for sure. All right. You can easily look at it, make those counts, and the same with this. All right. Only 2 colonies. All right. So as we look at each of these things and we're looking at our dilutions, it's a 10-fold difference as we're looking at these, and it sort of mirrors that as we look at this. So 2, I might expect maybe to see about 20-ish, all right, if I was really doubling or, 10 times more. Not exactly, all right. So 50, we're not exactly at 500 here, but pretty close ballpark, all right, with that. And that in turn, knowing some of this, you would want to have more than just one, right? So that you can kind of gauge this the littlest bit. But knowing what your dilution factor is ultimately allows you to go back and say, off of this 1 to 1,000 or 1 to 10,000, because that's easier to count, there must be X amount of bacteria in this, looks like full liter worth of broth. All right. Millions of bacteria. All right. So the pore plate, like I said, has agar. We typically call this stuff top agar. Usually it's already got agar in the Petri dish, and this goes on the top of it. In this instance, it's just straight up into the Petri dish. Doesn't really matter. All right. Your Petri dish would be sterile. All right. They come in plastic sleeves that have already been sterilized. And until you open up that lid, you should expect the inside of it should be sterile. And if you practice aseptic technique and carefully remove the lid, dump your stuff in, put the lid back on and swirl it up, should still be relatively sterile. All right. Only the stuff in your tube is what we want to be growing. So it's poured in there, swirled around. In this case, with a pour plate situation, we don't think about just a mil or 100 microliters that we put onto the plate. We dump the whole thing. So if that's got 10 mils total in it, the whole thing goes on there and we spread it around. And then look at after incubation for colonies. All right. And in these circumstances, I mean, for the most part, the bacteria is going to want to be growing kind of up on the surface. But you also sometimes can see some variation with that, with some microbes that might like a little less oxygen. They may be growing in the media instead of up on the top because of the way they were all mixed up in there. When you take bacteria from a broth and just spread them on the top of the plate, they just grow right on the top. All right. They don't sink in and start growing within, but here they're all dispersed. All right. So you may see some variation like that as well. This next idea is something I'm pretty sure you're going to do in the lab. The pour plating, not. All right. But the spread plating, the idea with this, here you will take a specific amount, 100 microliters. All right. And you pipette that and imagine that this has media on it. All right. You pipette a little of that and then you spread it around. All right. This glass spreading rod or hockey stick is what we tend to call it in the lab, allows the bacteria to be spread evenly. We sometimes use a little metal, a little cast iron turntable that you can spin. The plate spins around and you're moving your spreading rod just kind of back and forth as the plate spins, and it evenly disperses it. You can do it without that and just start spreading and turning on the bench top and get it all spread. All right. Same deal. After that, incubate it up, and there you go. All right. I will tell you this now, and I'm sure when you get to this point, the TA will also tell you this. These glass spreading rods, it's different than working with your inoculating loop. All right. When we go to sterilize the inoculating loop, you put that loop in the flame and you hold it there until it is red hot. You see it and you know, I've cooked whatever was on there. I've cooked it off of there, right? You don't want to take your glass spreading rod and put it in the Bunsen burner until it is red hot, all right? That is a bad idea, all right? You're probably also going to burn your hand, all right? So to prevent that from happening, we use alcohol, which is awesome, but it's also a potential fire hazard. You want to be careful, all right? The alcohol is in a little like jelly jar, quart jar, screw cap lid, but the little stubby guys, right? So not a quart jar, all right? If that catches on fire, which it could, you don't freak out. You're not going to destroy the building, although some of us would wish you would, so we could get a new one up in here. All you do is take your lid and put it back on there and screw it down, and the fire will go out. Fire needs oxygen. The screw cap jar, perfect. All right. Had a lab in Idaho Falls. They were all in Tupperware. That sucker caught on fire. So too did the Tupperware. And as it's all burning and melting and getting onto the bench, people are like, wait, my lab manual. That was an issue. I didn't get to see it. I rolled in right after the fire extinguisher had been used and put it all out, which was awesome. I thought the TA, he looked like he had just seen a ghost, right? He was very pale, probably pooped his pants a little bit, I'm thinking, right? Probably, because he's like, oh my God, everyone's gonna die, right? They did not, all right? So when you use that spreading rod, you've got the alcohol pretty close to your flame. You dip the glass rod into the alcohol, you pass it through the flame, and you move it away, not over your alcohol to the other side, all right? That glass rod likely is going to drip some flaming alcohol onto your bench top. That doesn't matter. They're fireproof. Maybe I shouldn't have told you that. They're fireproof, all right? Your lab notebook or any papers is not, so you want to be careful, all right? And as soon as that fire burns off of that glass rod, which is almost the second it goes through the flame, it's been sterilized. And that quick of burning and going away doesn't make it super hot. You quickly put it onto the plate, do your stuff, back to the alcohol, back through the flame, and now you've cleaned it off, just like you would do with your loop, all right? So you don't leave crap on it, right? Done deal. Dip, flame, whirl is the idea. Not flame dip. If you put it in the flame first and then touch it into the alcohol, you will catch your alcohol on fire. How do I know? I know. All right. So this one, this is an odd one. This is something that really until I started switched books and started with this one, this was, I think, the first time I saw this, and I was like, what the heck is this really even talking about, all right? I had to do a little further research to see what was up. So this uses some statistics to ultimately give us some ideas about what's going on. The book has some of this stuff spelled out at the end in one of the appendices. It's got a most probable number table that you could use to go through this, which I think is kind of interesting. All right. So in this case, we're trying to look really for coliforms, so fecal bacteria that could be, probably should be, in this pond water. You should probably expect there's going to be some of that. All right. So we've got some pond water in this flask up here, and then we pipette different volumes of it into these tubes. All right. So we've got 10 mils going into these, 1 mil and 1/10 of a mil. These are the same sugar ferment tubes, the phenol red broths that you guys used a couple weeks back. All right. You can potentially see the little Durham tube in here, and you definitely can see them down here. All right. So these have lactose in them. Coliforms, like our good friend E. coli, really likes that stuff. All right. So as we inoculate these, we can see we've got 5 positives here in the 10 mil. We've got 2 positives in the 1 mil, and we got nothing over here. All right. So this is like a code. How many of these tubes do we have? And then we go back to that table, and it, based off of what we see in our ferment tubes, tells us the amount that we've got going on here. All right. So similar, but a little bit different. All right. And as I saw that, I was like, well, that doesn't make sense, but it technically does. All right. As we're moving through there and incubating them up, it allows you to make those judgments as far as what's truly going on in there. Typically, when we think about like testing water, city water, or maybe you live out in the country and you've got well water and you're curious about it, you take in like a liter worth of that water and they run it through a filtration mechanism. The filter grabs the bacteria and they put that onto a petri dish and grow it up. Pretty sure you're going to do that toward the tail end of the lab. All right, a little bit of water testing. I think that's pretty cool. So I mentioned the spectrophotometer before, and using light to ultimately gauge how much growth are we really looking at. So we've got a spectrophotometer right here, and then we've got a couple of samples. All right. So our first sample up above is an uninoculated broth, a control, if you will. And this broth obviously has got some color to it. It's got stuff in there. It's yellowish, but it's pretty see-through. All right. So we make a reading with it to see what's the absorbance value on an uninoculated broth. And then deviations from that are going to allow us to extrapolate how much stuff is really in that inoculated tube. All right. So that's the bottom one. You can see turbid. All right. There's a lot of growth in here. So as the light is shining through this sample, as it's placed into the spectrophotometer, as it's moving through, some light moves through, is transmitted, and other light ends up getting absorbed or refracted off, but not moving through. The spectrophotometer can measure either way: absorbance, how much isn't going through, it's trapped by the sample, or transmittance, how much is actually going through. All right. Opposites of one another. All right. If you've got a high absorbance value, you've got a very low transmittance value. All right. Because hardly any light's making it through. It's all getting trapped in the super turbid sample. If you've got a very high transmittance, the absorbance is super low, not very turbid, right? Here you can see big thick line indicating basically all or virtually all of that light is moving right through the sample. And here, thick going in and very thin going out, very little getting transmitted, all right? So spectrophotometers make these measurements. And like I said, if we're thinking a growth curve, you'd be making repeated measurements over the course of the day, trying to see the change in this tube over the course of time. And then you could plot that and get some details about that. All right, how much growth is truly happening. All right, so when we think about the growth, typically bacteria replicate by binary fission, splitting in half. And when they do that, there are all sorts of potentials when we stain them up to think about some of the morphologies of that. How are these arranged? And we can see that with the gram stain, all right? Sometimes we see some variation, all right? Our cyanobacteria over here are filamentous. They replicate a little bit differently, fragmentation, breaking off, and we can see some little fragments in here. So like big long chains of cells, sort of same idea, but not exactly the same as binary fission, all right? This other one does it a little bit different. So we can see our little cell and larger cell, and these are budding, all right? So the N, not a nucleus, nucleoid, all right? So where we can see that, all right? So, and over here, same idea, all right? And I don't know about the nuclear envelope. That seems membrane-like. I don't know the details with that, all right? These are bacteria, all right? So budding off, this is something that a lot of yeast cells, when they're replicating, it's budding off mother and daughter cell, the daughter cell being smaller initially, all right? And these bacteria, that's the same, basically the same idea that's happening with them. So I'm pretty sure we saw this image in a previous chapter. Biofilms are ways that some bacteria may potentially grow. And they don't always have to be participating in some biofilm. They could be free living or free floating planktonic, or they may glue themselves down and become sessile, not moving, all right? So the idea with this, initially, some cells make contact, and this is a reversible attachment. So there is the potential that they may find this space and then decide, nope, and move away, all right? Or maybe they find it and they're like, okay, this is doable, all right? Maybe there's some nutrients and we want to stick around, all right? So initially it's reversible. They don't have to stay here, but once they get there, and if they are going to stay, technically it's just seconds before they're like, all right, I'm glued down. Now I'm here, all right? So pretty quick, all right, in this circumstance. And you can see the little flagella here with the potential, maybe we want to swim away, all right? So few seconds, now we start attaching. And these bacteria are those that have the capsule layers, the sugary coating on their surface makes it sticky, easy to adhere, all right? So after we decide, yeah, this is good, these colonizers ultimately have glued themselves down, irreversibly attached. And this may take a couple of minutes to get glued in place, if you will. All right. Then kind of lag phase sort of an idea. It takes them a little bit of time to get accustomed, and then they start growing. And we see cell division happening within this biofilm that's just established. And this can be hours to a couple of days, depending upon the respective organisms that are establishing in the first place. Once we get this kicked into gear and we're growing pretty well, we see a large colony, community of organisms, because oftentimes more than one type of bacteria may be participating in some biofilm situations, multiple things being utilized. All right. So they start, more of this sugary coating is being released. All right. The extracellular polysaccharides, and we can see more kind of community sort of situation with some channels, some water moving through there, and nutrients as well, providing cells that are deep within this access. All right. So again, more time within this period. And then we've got, I would say, a mature biofilm, if you will. All right. And others can come in. This is where we may start thinking more kind of community sort of stuff, other secondary colonizers. Once we've really got this stuff set up, others are like, sweet, could I come in? I could use some of your stuff and you could use some of mine, kind of mutualistic relationship. All right. And as we get all of this happening, after truly a good amount of time, we may start to kind of degrade the littlest bit within this sugary coating, allowing some of these microbes the ability to move away. All right. And we see some of these planktonic bacteria, dispersion of these new cells, and they ultimately will find a new place, settle down, and start to grow. All right. Is the idea. All right. Biofilms in your body, bad news. All right. It's very difficult to, if we're thinking like treating with antibiotics, super difficult to get the antibiotics in to that biofilm, to the inside where all of the cells really are, and we want to kill them. All right. We think of reoccurring types of infections, ear infections, sometimes are the result of some biofilm forming bacteria, and they kind of just keep happening. All right. And there's other stuff as well. So dependent upon who's participating in these biofilms, there are things that get secreted, molecules that get secreted that ultimately are communication molecules that allow these microbes to recognize situations within this. And it doesn't really even have to just be biofilm. They could be growing in a flask, and we could see some similar situations. So gram positives and gram negatives, little short peptides. We can see all these amino acids linked together, and then N-acetylated homoserine lactone, not the same, but serine is an amino acid. So positives with this, negatives with that. All right. And these quorum sensing is basically a mechanism that allows these bacteria to tell one another conditions are good, our populations are good, maybe let's disperse, or maybe some other situation. All right, where these are switches or triggers stimulating something within that culture of bacteria or within that biofilm to turn on or turn off if it's not not enough. All right. So quorum sensing with these molecules basically is like a switch, allowing them to recognize specifics, all right? Like time to disperse or something like that. So everything we've thought about, and almost everything really as we move through here and start thinking microbes, we oftentimes are thinking aerobic organisms. But anaerobic organisms are super important and definitely some that we could think about as disease causing. We think of these as really relatively primitive, but that's not really the best way to describe it. But ancient prokaryotes truly were anaerobic before we start thinking about oxygen revolution and all of that stuff. So there are opportunities to see anaerobic environments like this bog, or we could think about maybe in the gut of a cow. So in the rumen, the first stomach in the cow, very anaerobic, and different types of microbes can flourish in those situations. All right. So with our cow in the rumen with this anaerobic situation, there's a lot of methanogens that prefer this situation and other anaerobic bacteria. All right. This first stomach is where we start thinking about initiating a lot of the methane production that's coming out of the back end of this cow in the first place. Or first stomach, cows are ruminants. They are going to chew their cud. So what's in that first stomach is going to come back up and they're going to chew it up. So they could be belching out a lot of methane as well. All right. When we're thinking that first stomach. All right. So, and I think, when it comes to cows, I think this is really kind of impressive. As these cows are out in the pasture grazing, eating all sorts of grasses, cellulose, we don't digest it. We think of that as fiber. Cows, technically, they can't digest the cellulose either, but they get it digested by some of these bacteria in their gut. And they've got the enzyme cellulase that allows them to break it down. And in turn, then they release all of those glucoses and they're able to metabolize them. All right. I think that's pretty interesting. All right. So they speculate that before we ever settle down and start thinking hunter-gatherer kind of idea, our diets were really very different. Before we're like, holy cow, fire, we can cook stuff, right? We ate lots of plants, lots of leaves and things like that. And they speculate that our appendix that we now say is useless, probably harbored a lot of the same bacteria like we might think about in a cow that had the ability to break down the cellulose, and then we could metabolize all of those glucoses. And then as we shifted to a more starchy diet, amylase we produce on our own in our saliva, and amylase breaks down those starch molecules a lot simpler. And ultimately the appendix becomes kind of a vestigial structure, something that really the only thing it's useful for is for doctors to make some money as they rip it out of you, right? Probably. All right. Kind of crazy. So have you guys worked with thioglycolate tubes yet? No? Okay. I bet it's coming up pretty quick. So these, part of it makes me think sort of the Sims Deeps, but it's not exactly the same thing. All right. The Sims Deep, you can get some bacteria growing down in there that maybe are a little more facultative, so they can tolerate less oxygen. These tubes, the thioglycolates, and they will have screw caps on them, all right? The thioglycolates are a specialized type of media that basically establishes a pretty anaerobic environment, all right? Up towards the top, there's going to be a little bit of oxygen in that media. You've got some airspace in the tube, and a bit of the media has some oxygen. Down at the bottom, none, all right, or shouldn't be any, all right, if your tube is functioning like it should, all right. These tubes, and I think the ones we have a pink indicator for the presence of oxygen. There are others that are green. If that whole tube has changed colors before you inoculate it, you don't want to use that tube because it tells you it's full of oxygen, all right? A bit of pink at the top is okay. There should be a little bit of oxygen in there, but if it is all pink, bad news, all right? And you technically can take those tubes, crack the lid the littlest bit, put them into a boiling water bath, get them to a boil, and it will push the oxygen out with some gloves. You can grab those tubes, screw the cap back down, and they're usable. So just because they've changed colors doesn't mean that they're garbage. You can technically refresh them, all right? That's a little tidbit that you truly don't need, all right? So somebody will wind up in a lab working with those, and they're like, wait a minute, Dr. Shirley said we could do this. I'm going to try it, right? So all of these get inoculated, and then the cap's screwed on tight because you don't want oxygen really just going in there, all right? So we got a handful of different types of microbes up here that we're looking at. Obligate aerobes are first ones. They have to have oxygen. Obligate, you got to have it, all right? So they only grow up toward the surface, maybe even right up on the top of it, all right? And you don't see anything else. It's all clear, all right? And the littlest bit of growth or turbidity at the top. The exact opposite is an obligate anaerobe. These guys flat out hate oxygen and can't grow in the presence of it, all right? And you will only find them growing down at the bottom of the tube, as far away from the top as they can get. That's where they're going to be, all right? The facultatives, these guys, lots of bacteria fit into this category as a facultative anaerobe. They like oxygen. Technically, they prefer oxygen, but they also have capabilities of surviving in very low levels or maybe no oxygen, all right, and anaerobic type of environment. E. coli, a facultative anaerobe, likes to grow aerobically, loves it in those flasks full of media, shaking and plenty of oxygen. But your intestines aren't full of oxygen. Your intestines are a very anaerobic environment. And E. coli still is able to grow there. Not as good. It can't grow as rapidly as it can in the flask with ample oxygen, but it grows. And that's what this is really kind of trying to show you. Boatload of growth at the top, but we can still grow a little with very low oxygen. All right. So facultative. The aerotolerant anaerobes, these guys kind of are just like whatever, all right? You don't see great growth one place or another. It's pretty evenly dispersed, all right? So throughout, kind of this whole tube would sort of look turbid, all right? Aerotolerant anaerobe. And then the last one, these are the microaerophiles. So they like oxygen, but they don't like full oxygen, like atmospheric oxygen. They like it a little bit reduced. A little more CO2 makes them happy, all right? So you don't see them at the top. You see them just below the top where there's not full oxygen, but a little bit less. These are the microaerophiles, and they're kind of like the obligate aerobes over here, all kind of grouped up. They're grouped up, but the littlest bit below, all right? Some bacteria that we might think of as microaerophilic, we don't use these incubators, at least not in the teaching labs. But there are some incubators, just like you see as you walk in there and put your plates or tubes in there. Same looking type of incubator, but it's got a CO2 tank that pipes the littlest bit of carbon dioxide into the incubator, not making it flat out anaerobic, but reducing the amount of oxygen. All right. There are others, like our obligate anaerobes, that have the potential to be in an anaerobic chamber, no oxygen at all. And in situations, stuff we work with can tolerate being taken out and then put into another tube. It can be exposed to oxygen for a second, but some true obligate anaerobic bacteria, just grabbing it with your loop and pulling it out and putting it into the next thing, that little bit of air exposure killed it. All right. So they would be worked with in an anaerobic chamber that everything in there, it's glove box type of deal. So everything is free of oxygen and you cultivate them like that. I think we've got one of those that we'll see in just a second. Right here. It's like I knew. So this chamber, anaerobic chamber, this is where you'd put your hands, you do all of your stuff in there. Lots of tubes that you might inoculate, still have some airspace, but it's not oxygen. They use nitrogen or hydrogen and they pipe that gas into the tube and purge it out before anything is inoculated into it, ensuring that it's free of that. This other image, this is something that you will get to see in the lab. This is an anaerobic jar. There's a couple of different ways. This jar truly creates an anaerobic environment. There's a pouch in here that's got a catalyst. You pipette a little bit of water into it, seal the lid down, and as it starts reacting, all of the oxygen in this chamber gets destroyed, gets used in this reaction, and you're left with a CO2 environment, no oxygen, all right? Only anaerobes truly want to grow there, all right? There's another one that we use or sometimes use. We call it a candle jar. And it's like a big gallon pickle jar, a big gallon jar. You stack your plates in there just like this anaerobic jar. You stack them up in there. And then the top, before you put the lid on, you take a half a Petri dish, just the either, doesn't matter, and you put a little tea light candle on it. You light that candle and you screw the lid down. As the candle burns, it takes all of the most of the oxygen, not all of it, and then the candle burns out once the oxygen's depleted. Still a bit, not 100% anaerobic, but microaerophilic, all right? It dramatically reduces it down, all right? And you can see differences in candle jar versus anaerobic jar, all right? Clostridium is an anaerobe. It wants something like this, and giving it a microaerophilic environment, probably not the best. And you might get the hint, but probably not the best, all right? We should have started with this image. I'm just like, man, what a way to start your Wednesday, right? So here we're looking at a lot of necrotic tissue on this patient's foot, huge amount of edema. We've got redness. We got some nice toenails as well. That's just an aside. So there are lots of issues when we think about diabetes and stuff that's not being treated like it should, not dealing with it. And you can see a lot of neuropathy that can occur. You don't feel issues with your foot. The feet typically are the first to start with some of this neuropathy. So something happens and you don't even know, all right? And in turn, infections can happen and you also don't know, all right? There's other circumstances as we think diabetes and all of the sugar that's not being utilized like it should. That's perfect situation for some bacteria. They're like, sweet, feed me more, right? And I will grow and I will ferment, right? What we're really looking at is an anaerobe, Clostridium perfringens. Clostridium perfringens can cause gas gangrene. It is necrosis that we're looking at, dead tissue, all right? And my gut says, probably going to amputate this, right? Uh, Issues, all right? Oftentimes in diabetics, it may start with the toe and then the foot and then the leg and then maybe no more you, all right? Truly issues, all right, that can result with this. And once you start seeing necrosis like this, the potential for septicemia, for it to get into your bloodstream and move, huge issues, all right? So we'll get more details with that. All right, I just looked at my watch. I think it's quitting time. We almost wrapped all of this up. Catalase tests are where we will start on Friday. I'm pretty sure you've done these. Yes? Okay. Little bubbles. All right. Farewell. Have a great day.